You're listening Farmer Knowledge audio tracks. Please subscribe and click bell icon for increase your farmer knowledge.
Stability testing of new drug product. General selection of batches for stability testing. Test procedures and test criteria. Stability testing. Drug product. General, the design of the stability program for the finished product should be based on the knowledge of the behavior and properties of the drug substance and the experience gained from clinical formulation studies and from the stability studies on the drug substance. The likely changes on storage and the rationale for the selection of product variables to include in the testing program should be stated. Selection of batches. Stability information from accelerated and long-term testing is to be provided on 3 batches of the same formulation and dosage form in the containers and closure proposed for marketing. 2 of the 3 batches should be at least pilot scale. The third batch may be smaller, example, 25,000 to 50,000 tablets or capsules for solid oral dosage forms. The long-term testing should cover at least 12 months duration at the time of submission. The manufacturing process to be used should meaningfully simulate that which would be applied to large-scale batches for marketing. The process should provide product of the same quality intended for marketing and meeting the same quality specification as to be applied for release of material. Where possible, batches of the finished product should be manufactured using identifiably different batches of drug substance. Data on laboratory scale batches is not acceptable as primary stability information. Data on associated formulations or packaging may be submitted as supportive information. The first 3 production batches manufactured post-approval, if not submitted in the original registration application, should be placed on accelerated and long-term stability studies using the same stability protocols as in the approved drug application. Test procedures and test criteria. The testing should cover those features susceptible to change during storage and likely to influence quality, safety, and/or efficacy. Analytical test procedures should be fully validated and the assays should be stability indicating. The need for the extent of replication will depend on the results of validation studies. The range of testing should cover not only chemical and biological stability, but also loss of preservative, physical properties and characteristics, organoleptic properties, and where required, microbiological attributes. Preservative efficacy testing and assays on stored samples should be carried out to determine the content and efficacy of antimicrobial preservatives. Specifications. Limits of acceptance should relate to the release limits, where applicable, to be derived from consideration of all the available stability information. The shelf life specification could allow acceptable and justifiable derivations from the release specification based on the stability evaluation and the changes observed on storage. It will need to include specific upper limits for degradation products, the justification for which should be influenced by the levels observed in material used in preclinical studies and clinical trials. The justification for the limits proposed for certain other tests, such as particle size and/or dissolution rate, will require reference to the results observed for batches used in bioavailability and/or clinical studies. CPMP-IC. Any differences between the release and shelf life specifications for antimicrobial preservatives should be supported by preservative efficacy testing. Storage test conditions. The length of the studies and the storage conditions should be sufficient to cover storage, shipment, and subsequent use, example, reconstitution or dilution as recommended in the labeling. See table below for accelerated and long-term storage conditions and minimum times. And assurance that long-term testing will continue to cover the expected shelf life should be provided. Other storage conditions are allowable if justified. Heat-sensitive drug products should be stored under an alternative lower temperature condition, which will eventually become the designated long-term storage temperature. Special consideration may need to be given to products which change physically or even chemically at lower storage conditions, example, suspensions or emulsions which may sediment or cream, oils and semi-solid preparations which may show an increased viscosity. Where a lower temperature condition is used, the 6 months accelerated testing should be carried out at a temperature at least 15 degrees Celsius above its designated long-term storage temperature, together with appropriate relative humidity conditions for that temperature. For example, for a product to be stored long-term under refrigerated conditions, accelerated testing should be conducted at 25 degrees Celsius plus or minus to degrees C 60% RH plus or minus 5% RH. The designated long-term testing conditions will be reflected in the labeling. An expiration date. Storage under conditions of high relative humidities applies particularly to solid dosage forms. For products such as solutions, suspensions, etc., contained in packs designed to provide a permanent barrier to water loss, specific storage under conditions of high relative humidity is not necessary, but the same range of temperatures should be applied. Low relative humidity, example, 10 to 20% RH, can adversely affect products packed in semi-permeable containers, e.g. solutions in plastic bags, nose drops in small plastic containers, etc., and consideration should be given to appropriate testing under such conditions. Conditions minimum time period at submission. Long-term testing 25 degrees Celsius plus or minus to degrees Celsius per 60% RH plus or minus 5% 12 months. Accelerated testing 40 degrees Celsius plus or minus to degrees Celsius per 75% RH plus or minus 5% 6 months. Where significant change occurs due to accelerated testing, additional testing at an intermediate condition, example, 30 degrees Celsius plus or minus to degrees C 60% plus or minus 5% RH should be conducted. Significant change at the accelerated condition is defined as 1. A 5% potency loss from the initial assay value of a batch. 2. Any specified degradant exceeding its specification limit. 3. The product exceeding its pH limits. CPMP ICH3A batches. The overall shelf life may depend on the minimum time a batch may be expected to remain within acceptable and justified limits. The nature of the degradation relationship will determine the need for transformation of the data for linear regression analysis. Usually the relationship can be represented by a linear, quadratic or cubic function on an arithmetic or logarithmic scale. Statistical methods should be employed to test the goodness of fit on all batches and combined batches, where appropriate, to the assumed degradation line or curve. Where the data shows so little degradation and so little variability that it is apparent from looking at the data that the requested shelf life will be granted, it is normally unnecessary to go through the formal statistical analysis, but only to provide a justification for the omission. Limited extrapolation of the real-time data beyond the observed range to extend expiration dating at approval time, particularly where the accelerated data supports this, may be undertaken. However, this assumes that the same degradation relationship will continue to apply beyond the observed data, and hence the use of extrapolation must be justified in each application in terms of what is known about the mechanisms of degradation, the goodness of fit of any mathematical model, batch size, existence of supportive data, etc. Any evaluation should consider not only the assay, but the levels of degradation products and appropriate attributes. Where appropriate, attention should be paid to reviewing the adequacy of the mass balance, difference stability, and degradation performance. The stability of the drug products, after reconstituting or diluting according to labeling, should be addressed to provide appropriate and supportive information. Statements labeling a storage temperature range may be used in accordance with relevant national regional requirements. The range should be based on the stability evaluation of the drug product. Where applicable, specific requirements should be stated particularly for drug products that cannot tolerate freezing. The use of terms such as ambient conditions or room temperature is unacceptable. There should be a direct linkage between the label statement and the demonstrated stability characteristics of the drug product. Annex 1. Glossary and Information. The following terms have been in general use, and the following definitions are provided to facilitate interpretation of the guideline. Accelerated testing studies designed to increase the rate of chemical degradation or physical change of an active drug substance or drug product by using exaggerated storage conditions as part of the formal, definitive, storage program. These data, in addition to long-term stability studies, may also be used to assess longer-term chemical effects at non-accelerated conditions and to evaluate the impact of short-term excursions outside the label storage conditions, such as might occur during shipping. Results from accelerated testing studies are not always predictive of physical changes. Active substance, active ingredient, drug substance, medicinal substance. The unformulated drug substance, which may be subsequently formulated with excipients to produce the drug product. Bracketing. The design of a stability schedule so that at any time point only the samples on the extremes, for example of container size and or dosage strengths, are tested. The design assumes that the stability of the intermediate condition samples are represented by those at the extremes. Where a range of dosage strengths is to be tested, bracketing designs may be particularly applicable if the strengths are very closely related in composition. Example, for a tablet range made with different compression weights of a similar basic granulation, or a capsule range made by filling different plug fill weights of the same basic composition into different size capsule shells. Where a range of sizes of immediate containers are to be evaluated, bracketing designs may be applicable if the material of composition of the container and the type of closure are the same throughout the range. Climatic zones. The concept of dividing the world into 4 zones based on defining the prevalent annual climatic conditions. Dosage form, preparation of pharmaceutical product type, for example tablet, capsule, solution, cream, etc., that contains a drug ingredient generally, but not necessarily, in association with excipients. Drug product, finished product, the dosage form in the final immediate packaging intended for marketing. Excipient, anything other than the drug substance in the dosage form. Expiration date, the date placed on the container labels of a drug product designating the time during which a batch of the product is expected to remain within the approved shelf life specification if stored under defined conditions, and after which it must not be used. Formal, systematic studies. Formal studies are those undertaken to a pre-approval stability protocol which embraces the principles of these guidelines. Long-term, real-time testing. Stability evaluation of the physical, chemical, biological, and microbiological characteristics of a drug product and a drug substance, covering the expected duration of the shelf life and retest period, which are claimed in the submission and will appear on the labeling. Mass balance, material balance. The process of adding together the assay value and levels of degradation products to see how closely these add up to 100% of the initial value, with due consideration of the margin of analytical precision. This concept is a useful scientific guide for evaluating data, but it is not achievable in all circumstances. The focus may instead be on assuring the specificity of the assay, the completeness of the investigation of routes of degradation, and the use, if necessary, of identified degradants as indicators of the extent of degradation via particular mechanisms. Matrixing the statistical design of a stability schedule so that only a fraction of the total number of samples are tested at any specified sampling point. At a subsequent sampling point, different sets of samples of the total number would be tested. The design assumes that the stability of the samples tested represents the stability of all samples. The differences in the samples for the same drug product should be identified as, for example, covering different batches, different strengths, different sizes of the same container and closure, and possibly, in some cases, different container closure systems. Matrixing can cover reduced testing when more than one variable is being evaluated. Thus, the design of the matrix will be dictated by the factors needing to be covered and evaluated. This potential complexity precludes inclusion of specific details and examples, and it may be desirable to discuss design in advance with the regulatory authority, where this is possible. In every case, it is essential that all batches are tested initially and at the end of the long-term testing.